Welcome everyone to session 3, which is enabling technologies. Hopefully you've had a chance to view the talks. There was one technical glitch on the first talk on bioprinting. There was a little bit of issue with loading, originally loading the talk. So it's now available. So if anyone wasn't able to view the talk, they can go back and view it now. And I'll be happy to answer any of specific questions on the talk through the messaging through the app during this meeting. Okay, so everyone's met all of the speakers. This is the live panel discussion part for enabling technologies. So we welcome all of your questions. You can either type them out in chat, and I think I'll just start with a couple of questions for Dr. Anderson. You talked about the lipid nanoparticles, and I know a lot of people are intimately familiar with them with the vaccines that a lot of us have taken. One of the issues has been storage. Going forward, how is that going to be addressed in terms of temperature and shelf life?
Yeah. Well, I think one clear direction is that formulations are moving to a lyophilized state. There are a number of mRNA vaccine formulations that I'm aware of in development that are already lyophilized, and that really dramatically improves the shelf life. So I think that's probably the most important advance. And whether or not that affects all LNPs in the future, I'm not sure, but clearly some are going to be dry powders.
All right, thank you. Dr. Marini, with your imaging talk, you gave a very nice presentation of the number of types of imaging, including the optical tissue clearing. What about, could you just address briefly the real-time in vivo imaging?
Yeah, sure. Yeah, I mean, that's another enabling technology that is used a non-destructive. It is used frequently to monitor and observe pre-labeled cells that have been injected into an animal of interest. Can you hear me okay?
Yes. Yep.
Great. It's normally used or frequently used using cells that have been pre-labeled, stem cells that are pre-labeled, or tissue construct that's been pre-labeled. And these pre-labels can be anything from a light-emitting enzyme, such as luciferase, to a fluorescent nanoparticle that's been incorporated into the cell. And there's a few ways of accomplishing it. The light emitters can be monitored using a really sensitive CCD camera, and then the collection of photons is overlaid on top of usually some form of a black and white image of an animal. Alternatively, there are ways of putting microscope objectives into these animals, and you can actually monitor fluorescent particles using kind of a standard microscope objective, normally in some form of a multiphoton or these new laparoscopic stick objectives where they can be placed in small incisions in the animals. Yeah, I mean, non-invasive in vivo imaging is really a great way of tracking in real time stem cell migration, tissue incorporation. Depending on the cell type, you can label pretty much anything. And there are multiple methods to label different wavelengths of light, or I should say different emitters of different wavelengths of light, allowing you to monitor multiple populations of cells at the same time.
Oh, thank you. There on the chat, there are a few questions showing up now. Again, for you, Dr. Marini. I'll go ahead and read the question. Would TIRF microscopy be of any benefit in imaging of tissues after the clearing process?
Sure. Total internal reflectance microscopy is actually, so is it at any of interest? You can do it then. However, a lot of the lipid membrane has been removed. Total internal fluorescence has been used primarily to look at proteins docked in the lipid membrane. The total depth of field resolution is on the order of single nanometers. It's pretty, sorry, single microns. It's pretty short, but using the tissue clearing, it actually allows all the proteins to be fixed in three-dimensional space. So you can still do turf type imaging and see the proteins that are docked at the membrane surface, but the actual lipid themselves most of them have been removed. It's a great new technique, requires novel microscope objectives, but it does allow you to address a lot of questions of interactions of all the different cells, receptors, and all the signaling proteins associated with receptors. Really interesting technique.
Right. Thank you. There's also a question from David Turacek for Dr. Anderson. What controls the homing and circulation of nanoparticles given they are not alive and not capable of chemotaxis?
So one main determinant of nanoparticle homing is the surface chemistry. And a good example of this is for the drug Onpattro. What we know is after injection, the lipid surface becomes associated with something called ApoE, which is normally used in the body to help with trafficking of dietary lipids. So now you have a nanoparticle coated with ApoE, and hepatocytes in the liver have an ApoE receptor, and that then leads to selective uptake in those tissues. It's less well understood in targeting other tissues without the presence of ligands, but I think the leading theory is that other, differential absorption of serum factors can lead to nanoparticles that ultimately accumulate in different tissues.
Thank you. Let's see, we're getting questions in now. Here's a follow-up question for Dr. Anderson. Are the LNPs, even if targeted, also taken up specifically by the reticular endothelial system and or phagocytic cells?
So, you have a population of nanoparticles injected in the blood. Depending on their chemistry, there will be some accumulation in phagocytic cells. How much of the nanoparticles go to those, say, macrophages or monocytes versus the destination tissue really depends on the chemistry. But it is common, since those APCs are natural scavenging cells, for nanoparticles to end up in them.
Right. Thank you. There's a question for Dr. Musunuru. Were there any side effects of gene editing you observed in monkeys during the long-term, the 238-day study? Examples of loss of weight, et cetera.
Yeah, thanks for the question. So the only arguable side effect, if you will, was a moderate rise in transaminases, so AST, ALT, in the first week after receiving the lipid nanoparticle treatment at day 0. And that's expected with an LNP, with LNP drug. Besides that, we have not seen any side effects either in short-term studies or in the ongoing long-term study. No adverse clinical signs whatsoever, besides that transient rise in the transaminases that goes away within 1 to 2 weeks. They seem to be entirely healthy, no clinical signs whatsoever. So it seems like the LNP treatments have been extremely well tolerated.
Thank you. There is a question for Dr. Marini. Can the relatively new digital holographic microscope be used on 3D organs or only on monolayers?
Yeah, thank you, Chris. I think this is a planted question since we just bought a phase holography microscope last week. Um,
I didn't realize that we got that. I found out by accident because Rasa and I had been
okay.
reading about that and
Yes.
said, wow, that sounds really cool. I wonder what we can do with that. And someone
Right.
told us, well, you know, we just bought one. Wow.
Right. So yeah, so what is it? So essentially it is phase contrast microscopy with a baseline of a laser run through it. And the resulting laser is scanning in a single dimension and then allowing reflectance and other things like off-axis Fresnel Fourier transformations. You can do phase shifting. And it gives you so much more information from a single phase contrast as it is actually monitoring the surface of a cell. So from what I know, it is currently used on monolayers and also for cell tracking. But one of the reasons we bought it is we want to see if it works on organoids. And that work has not been done yet. So I don't have a direct answer for that. But what it does do is give you a tremendous amount of surface information on individual cells. We can differentiate cells now basically on the shape of the cell on plastic or a matrix. And also, as the cell is migrating, it has a very characteristic movement pattern, which is also identifiable. So absolutely fascinating technology. I'm looking forward to exploring it more.
Can I ask just a follow on to that? So part of the reason why I had been reading about that was because we were preparing a proposal for NASA. Yes. And we were trying to figure out what kind of imaging we could do on a random positioning machine. And apparently it turns out almost none because all of the imaging is sensitive to vibration. And I read one paper at least that said that this particular technology is completely unaffected by the vibration. So I guess the investigators have actually designed a portable system that they could put on the random positioning machine with their cells to kind of image them in real time as they were being exposed to zero gravity. So
what's absolutely fascinating is there is actually two laser lines on this system, one that scans the cells and one that stays perfectly in tuned. And it actually monitors shifting of motion and subtracts it from the actual baseline itself. So yeah, absolutely fascinating technology. And it does, we've asked the field technician that came in to install it, whether or not we needed a really expensive vibration-free table for it. And he's like, no, it's insensitive to that.
All right, thanks. There's one question for bioprinting. Would you see an advantage in developing a bioprinter with a print head on a 6-axis robotic arm? And the ones that we've developed at WFirm and you can buy commercially are primarily X, Y, and Z axis bioprinters. But there are some even commercial ones that use a robotic arm, which gives you much more freedom in terms of how and where you're going to do your printing. So I think as we move forward, these types of robotic arms will be used more and more in terms of bioprinting. You know, ultimately, it would be nice to go in, say with a Da Vinci, and to do surgery, and then you go at the same time, you can print where you've taken out tissues and print right at the site of surgical removal. So those types of print heads will be critical as we move into the future. Okay. All right. One question for Dr. Parada and Montanaro is very nice describing use of gene therapy and gene editing. What do you think the future holds for gene editing? I know you mentioned a number of different clinical trials that are ongoing now using gene therapy. What do you see for the future? For either Dr. Parada or
I can go first, I guess.
Yep.
So you're asking more about gene editing, I'm guessing.
Right. Right.
Therapy.
Yeah, more gene editing. Yes.
Okay, so gene editing. So Dr. Anderson in his talk mentioned that the company Intellia has a clinical trial underway using lipid nanoparticle delivered CRISPR-Cas9 to target the transthyretin or TTR gene. So they started dosing patients late last year. And in fact, just last week, they announced that they're going to be reporting the first results from that phase 1 trial on the 26th of this month. So in a couple of weeks, we will know how well it worked, or hopefully get some indication of how well it worked, even though phase 1 trials are typically more about safety than efficacy. So that's exciting. I think many of you are aware that CRISPR-Cas9 is currently in clinical trials with sickle cell patients, and we've been receiving updates on that for more than a year now. And 22 patients have been treated as of the last update, which was, I guess, late last week. And it seems that they are, by and large, doing well. And, you know, maybe it's a little too early to be saying the word cure, but it sure does seem like they're no longer having the sickle cell attacks or the need for transfusions or things that typify sickle cell disease. So at least functionally, they do seem to have substantially improved. And if that persists, you know, for more than a year or so, the first patients have been studied so far, then they may well have been cured. And that's very exciting. There are other trials underway for CAR T immunotherapy using gene editing to develop better CAR T cells for a variety of immunotherapies. So there's a lot of activity in this space. There's another one for a genetic form of blindness. We haven't heard the first results of those yet, but I think we can expect them later this year. And so with both rare diseases as well as more common diseases like cancer and cardiovascular disease, I think the next few years are going to see a flood of interesting results, hopefully positive results. And you know, in the long run, I expect it's going to transform the practice of medicine. I don't think I'm understating it, or overstating when I say that.
Right. I think you're right on that. Yeah. Thank you. Chris, do you have anything to add, or?
No, I think that covered pretty much everything. I think. Yeah, that was,
right.
Can I ask a quick question to Dr. Musunuro about the sickle cell? So this was hematopoietic stem cells that they took out autologous, okay, and edited and put the cells back. Okay. Do you see any difference in? So that to me would seem to be a little bit lower hanging fruit, I guess, than actually delivering in vivo the editing machinery, because you have control of which cells you're working on, and you can actually check them to look for off-target effects and things like that before you put the cells back.
Exactly. There's one company so far, well, a team of companies, CRISPR Therapeutics and Vertex, that has made it into phase 1 clinical trials with editing solution for sickle cell. Exactly as you said, that was ex vivo, so hematopoietic stem cells taken out of a patient, the editing done outside of the body, and then those cells return to the body. And that, of course, requires some degree of myeloablation, you know, the conditioning and so forth. So there's some toxicity associated with that. And so you're exactly right. The ultimate goal would be to be able to deliver the gene editing tool, perhaps with something like lipid nanoparticles directly into the body, targeted to the hematopoietic stem cells within the body, so that the end takes place within the body. And that would be so much more elegant and so much more better tolerated by the patients. And although we're quite a ways from human trials, at least a couple of companies have now publicly reported data showing in mouse models that they have been able to achieve some degree of in vivo targeting of hematopoietic stem cells at the sickle cell locus or otherwise, rather the beta hemoglobin locus or otherwise, which seems promising. I think, you know, given enough time and investment, we will get there where diseases like sickle cell that you traditionally think of as being ones where you're transplanting cells, taking cells out and transplanting them back in, will become in vivo solutions.
Thank you. So I just noticed that we're right at 4 o'clock. So I think it's time to actually end the session panel discussion. I'd like to thank all of the speakers for giving excellent talks today, and for everyone that attended the panel discussion and asked questions. Again, thank you, and hopefully see you during the rest of the meeting.